Hi, I am Uwe Hilgert, and I'm joining you today from Tucson, Arizona. I am a professor in the Bio5 Research Institute at the University of Arizona here in Tucson. My research interests are in microbiology, genetics, and in bioinformatics, and also in STEM education. I also teach, and I used to work for many years in Cold Spring Harbor with a great team in the DNA Learning Center. Thank you for joining me today for a bioinformatics laboratories in which we will analyze phage Lambda DNA via electronic restriction analysis. Questions? Well, use the chat. You can also send me an email at my last name, hilgert@bio5.org. Just give me a couple of days to respond, please. If you want to work along, please make sure that you're using a laptop or a computer. A tablet might work. The worksheet that we'll be working through this exercise will be available at the Resources tab at the DNA Live website. And one of our tools is actually requiring Flash software. Through this computer lab, you will now understand how bioinformatics enables us to understand how life works. Organisms store information in DNA and proteins, and computers enable us to decipher that information. The field of bioinformatics is the study of how living organisms use nucleic acids and proteins to store and process information. In turn, as we better and better understand how life handles information, it will help us to also design better information technology, computers and software, not only in biology, but also in finance, healthcare, civic administration, and many other subjects. Nucleic acids store information in the sequence of the nucleotides, just as we use language, letters, words, and sentences to communicate and store information. Now, DNA only uses 4 letters to design basically any life form, from animals to plants to bacteria to fungi and so forth, while in the English language, we need 23 letters to just talk to each other. But if you look at it, Morse code and computer language also utilize 2 elements each, the zeros and 1 in computers and the beeps and dashes in Morse code. So DNA is comparatively luxurious, having 4 letters available. DNA is like a hard drive. It stores information but doesn't do anything. Proteins are life stewards. A few molecular basics bullets here. A gene is a segment on the DNA that encodes a protein. DNA is transcribed into RNA, and from that information can flow into proteins via a process called translation. And here you see an image of the central dogma of molecular biology. DNA is transcribed into RNA, is translated into protein. As far as the elements are concerned that these languages use, DNA and RNA both use 4 letters. That's why this is basically a transcription from DNA to RNA. And then proteins, in order to do all the functions that they can do, use 20 amino acids. So a much richer information molecule, but they have to do so much more. They're not only storing information, but they're digesting starch in our guts, they're sensing light in our eyes, proteins do all types of stuff. To get from the 4-letter language of DNA and RNA to the 20-letter language of amino acids, there is a whole genetic code necessary whereby 3 nucleotides place 1 amino acid in a protein. So if you look at the genetic code from the perspective of DNA, where you have a TTT in DNA, you would have an UUU in RNA, which would lead to the placement of phenylalanine in a protein chain. And there is a 1-letter symbol also for that amino acid, that's an F. Another one that we could look at is what is also called the start codon, ATG, down here. ATG, or on the RNA, AUG, places a methionine in the amino acid chain. And in fact, it also starts every amino acid chain, every protein, with the amino acid methionine, which can be abbreviated with an M. We're going to talk and focus a little bit on viruses and on specific viruses that actually infect bacteria. And this little buddy here is an electron microscopy graph of a T4 phage, which consists of a head that contains all the genetic information, a tail, and tail fibers. The tail fibers conduct the connection to the bacterial victim, and the head is where the nucleic acids are stored. Let's look for a moment at viruses and phages. What do viruses do? In this area where we talk a lot of what all the bad things that coronaviruses do, it might surprise you if I tell you that viruses don't do anything. They are particles that hold the genetic information that host cells would utilize to make more viruses. So what I'm saying is that we are, as in our cells, if a virus attacks our lung cells, for example, we are basically utilizing the genetic information of the virus to make more virus in those lung cells that get infected. Are viruses alive? Oh, let's talk about that for just a moment. What are the hallmarks of life? You have metabolism, picking up food, metabolize, like digesting it, extracting the energy, extracting substances, molecular components to build our own bodies, and ultimately excreting what we don't use. We have, as another hallmark of life, the capability to interact with our environment, to react to stimuli, to provide stimuli, right? And maybe a third hallmark here is that we are able, on our own volition, to procreate. For single-cell organism, that means that they conduct cell division, and for animals and plants that procreate sexually, you know, we are able to do all that on our own. Viruses cannot. They need to infect us so that we do that work for them. And bacteria have viruses too. As we have diseases, they have problems too, potentially from viruses. And viruses that infect bacteria are called bacteriophages, or for short, phages. Let's watch a movie together. This 3D animation depicts bacteriophage T4 and E. coli bacteria. It is based on scientific research. Please watch closely and try to identify the different steps in the infection process. Later, please enter your observations into your worksheet. There's no narration, and I'll shut up now too. So here's our worksheet, and the first part of it is only repeating the information that's on the DNALC Live website. So let's scroll down to the second part where the real worksheet starts. And we worked actually through some of that stuff already here, what are the hallmarks of life and so forth. So let's move on to the next one, because we have seen already a part of the true phage attack, and that's just the first third of what really all goes into a phage successfully infecting a bacterium and then prompting the bacterium to produce more phage and then to release them into the environment. What I want us to do is I want us to actually dissect all these different phases of the phage attack here, and we're going to do this in more detail than what we had seen in the video. If you have printed this out or have otherwise a piece of paper, I want you, through the next series of slides that I'm going to show you, actually start to maybe write some of these steps down that are significant for the phage attack. All right? So let's go back to our slide presentation. Yes, we were at the worksheet. And this is just giving you an idea of the size proportions between Escherichia coli cells and phages. So phages are significantly smaller, but they are not that small that they would be just dust specks on the surface of a bacterium, right? And you see the scale down here, that bar is 1 micrometer. And let's move on and look at the infection again. So we saw this kind of like moonlander capsule here, drifting basically through the medium and bumping into a bacterium. So they don't swim actively towards bacteria, but bumping into a bacterium. And then as far as the bacterium is concerned, let's look at that for just a moment again, as far as what we saw in the movie. Unfortunately, my movie, I must have closed it entirely. So let me open it here again. Here it is. And we're not going to play the entire movie again. We're just going to stop it here and then seeing what's happening. The phage is bumping into bacterium and then anchoring in some very specific structures here that don't resemble just the surface of the bacterium. It's much more complex than that, right? And then even the small fibers, after the long tail fibers have attached, the small fibers have their own structures that they attach to, right? So this adsorption phase consists of 2 steps, and then the phage goes ahead and uses that very solid anchoring to actually how to say, extend the tube through the bacterial membrane. So let's come back to our PowerPoint presentation here. So that first phase is called absorption, and there are 2 phases, the large fibers attached, the small fibers attached, right? Then we have that extension of the tube through the membrane and the injection of the DNA into the bacterium, and that wasn't shown in the movie at all. That movie only pertained to the work that the research laboratories did that we saw in the acknowledgements. And this DNA then is utilized by bacteria to do what? Well, it's the phage DNA, so they will be phage components. Through transcription and translation, they will build phage proteins and start assembling them in heads and things and tails and small and large fibers. And on the other hand, we have here the bacterial system that replicates the phage DNA as well. So it's not just making phage protein, but also making more phage DNA, right? In the next cycle, this will all then be assembled into the next step. It all be assembled into complete phages and more phages until finally the bacteria lyse and release all these phages into the environment. Now, what that lead to? That leads to a situation where actually originally one phage affected one bacterium. The bacterium produces more phage and they can bump into the bacteria around and basically read that instruction through the entire system, right? So let's go back to the worksheet. As impressive as that all was, let's go back to the worksheet, and I hope you started writing in here, right? So let's say large tail fibers attach or absorb. Well, let's call it attached because ultimately it's the absorption is both together, right? The large and the small. Small tail fiber attached to specific structures and not just somehow sticking to the surface. No, that sticking is very well mediated by those specific structures that are on the bacterial membrane and to which the phage fibers fit. So I'm not going to fill this all in by hand now. I did that work actually beforehand. And so here is actually a sheet that shows you the responses. That is not available on the drive that I showed you, so you can't download this. But it's basically what I hope you already took notes. There are the steps that we could observe, right? The tail fibers attach, the phage contracts and it extends its injection tube that breaches the bacterial host membrane. Then the phage injects genetic material into the bacterium. The bacterium expresses, and that's again transcription and translation, the phage genetic information. I could have written here also a host replicates bacterial DNA. And actually, I'm not going to do this right now, but that's a good idea. Then proteins self-assemble into phages, not just phage proteins, but into entire phages, and let's fix this here because, you know, it's a Word document. into entire phages, and then ultimately the host is being lysed, and the new phages are all released to infect other bacteria. Okay, so I hope you have some of these steps in your assortment, and I want to bring you now back to the PowerPoint, because there's something really astounding that happened. So we understand now phage attack on bacteria, and yet research scientists in the early 60s found situations that actually, despite the presence of phages, there were E. coli strains, E. coli species, that could not be infected. E. coli lived on, but how so? Let's go back to the worksheet. How so? The next, after we've done now all these different steps in the infection process, I want us actually to think about how could a bacterium develop immunity to each of the different phases of the phage attack. And here's why this actually is important. This is not just because some bacteria develop immunity to bacterial viruses. It's also important for two very, very significant reasons. The first one is, I want you to actually start thinking like a biomedical researcher that tries to develop treatments, that tries to develop prevention mechanisms to actually potentially disable that virus can affect us, right? And phages and viruses is sort of the same thing. The steps are very, very similar. Some viruses may not have fibers, they may have other elements that can attach. The coronavirus is somewhat different. It's not called fibers on the coronavirus surface. However, the thinking of like, how could we interfere with any of these different steps that we just figured out in the responses sheet here as it goes along? And let's first think about the fibers attaching. What could prevent that? Remember how the fibers attach to very specific structures on the bacteria? Those structures are proteins. The proteins are encoded in bacterial genes. If those genes experience mutations that would change the proteins so that the phages can't actually attach anymore, they would either, as far as the long fibers are concerned, not even get to hook onto the bacterium, or even if that's possible and the mutation only happened in the genes for the structures for the small fibers to attach, then the phage wouldn't have this secure hold to actually start compressing and extending that tube, right? So this kind of a mutation could yield that actually, as far as the fibers are concerned, that the structures are just not there on the bacterium surface anymore, that the fibers would attach to. Now, the phage contracts, so it has actually some time on the surface of the bacterium. If the bacterium had a mechanism developed in its mutation, right, that could actually scrub the surface and remove phages that are sitting out there, there would be no place that the phage could extend its tube into, because it would already be on its way again, right? Further, the phage extends the injection tube and breaches the bacterial host membrane. Well, can you imagine if the bacterium had a mutation that would actually fortify the outer layer to the extent that that tube could just not penetrate? The phage attack would be over right there. Now, if all this still worked fine, but the mutation would be at a later point, let's say the phage can extend its tube in the bacterium, but maybe the bacterium has learned to clog that tube so that the DNA couldn't come through, right? A different protein just clogging that tube, fine. Or if the DNA still gets inside, maybe destroying the DNA. The phage injects its genetic material. If this could be destroyed by means of a protein that can do that, there would be no success for the phage at all. Finally, if you get that genetic material still into your system as a host bacterium, or as our lung cells for corona, yes, good example, then if we were able to actually somehow disable the transcription and translation of the phage genetic information, or the replication of the phage genetic materials in our cells, right, that would stop it right there. Or if we become able to synthesize a protein that would actually disable the assembly of the virus before it leaves our cells. And then finally, something that might not help ourselves, but it would at least help the community. If you think about that, let's go back here, would at least help the community if in this step here, in this step here, we could prevent lysis, that our cells, our lung cells for that matter, or the E. coli cells for the, remember, they found immune E. coli, that it would actually, yes, sort of not being a real E. coli cell anymore and not being able to do anything, but if it had a protein that would fortify its exterior, not to the extent that it prevents phage attachment and tube extension, but to the extent that it's almost like a net around, right? That the bacteria couldn't blow apart. Then the phages wouldn't be released and infect other lung cells. I mean, other E. coli, or in case of corona, other lung cells, right? So there is actually quite a number of possibilities that you could think of that you could write into your responses here. And I have them all in this response sheet. And I want to go back to the PowerPoint with you guys. So we're coming back to this slide where E. coli was surrounded by phages. There were enough phages available to kill E. coli and infect it, make more phages and so forth. But E. coli lived. So this kind of immunity triggered the question, how so? And led actually to a discovery which laid the foundations for molecular cloning and genetic engineering. So researchers found that actually the phages were still able to absorb to the E. coli that were immune. They found that the DNA could be injected, but the next thing that E. coli did was then that the DNA was degraded by proteins that E. coli synthesized, so-called restriction enzymes. Now, the term restriction might mean that they restricted phage attack, or that they restricted the DNA, just another word for cutting, and leaving the DNA in shambles, in fragments here, right? The phage DNA. Okay. So further examination showed that those DNAs were actually not just some DNAs that degrade DNA willy-nilly, but that they were very specific in, especially the first one that they found, Escherichia coli restriction enzyme 1. They found that it actually degrades DNA at a target sequence that is GAATTC, right? G followed by A, followed by A, followed by T, followed by T, followed by C, which, of course, on the other strand would then be, again, GAATTC. Basically the same sequence in one direction, in the other direction, right? And if you remember some of the work that you did potentially in a restriction analysis laboratory, DNA has a directionality, 5', 3', and then 5', 3' on the reverse strand. Now, researchers found also that DNA was not just cut bluntly, but that the cuts occurred between G and A. And what does cut mean here? It means basically that when this sequence was present in the target DNA, that the enzyme ECORA1 was harping in on that sequence and then destroying the sugar phosphate backbone between these two nucleotides. And the same happening here. So again, not a blunt cut, but a somewhat staggered cut, so that when you imagine that these two fragments would now fall apart, because all that would hold them together is the hydrogen bonds between the two A's and the two T's here, that you would actually have some sort of overhangs, right? There would be an overhang to this whole fragment consisting of those 4 nucleotides and this fragment consisting of those 4 nucleotides. And shortly after, researchers in another laboratory found a restriction enzyme in Haemophilus influenza. And my hunch is it wasn't the first one they found, because they called it HIN, for Haemophilus influenza, D3. And that enzyme cuts very similar to ECORA1 in that staggered kind of situation, with the difference of the target sequence, right? So it has a total different target sequence than the ECORA1 enzyme. Now, how did this lead to genetic engineering? Well, so first question to you is, can you find the GAATTC in the sequence up there? That's a tough one, right? Let's just let an enzyme do the job. All right. Those enzymes are maybe a little bit more cognizant to stuff than we are. So the enzyme found and identified the GAATTC and administered those cuts to it. And there was actually 2 sites here, right? Leaving us with 1, 2, 3 fragments, if you wish. And those fragments then would basically fall apart, because these weak hydrogen bonds here, no more sugar phosphate holding these 2 strands together, would lead to basically 3 fragments that would drift apart or fall apart, however you want to call it. And then, but ultimately, these sticky overhanging ends could then lead to a reassembly if there is an enzyme present that can actually reconnect the G and the A and the G and the A, right? And those enzymes exist in nature too. So how is this being used for genetic engineering? Well, let's go ahead up. Let's look at this one more time so that you see this actually with a different little bit color here indication. So you see actually how this middle fragment falls off, right? You could actually imagine that this can also go the other way, that if you had two sets of sticky ends that are complementary to a piece of DNA that you would add to this mix of those two, that it could actually be inserted and therefore clone a piece of DNA in, say, a cloning vector, in a cloning DNA tool. How would that lead itself to genetic engineering? Here you see a photograph of a jellyfish. And that jellyfish carries in its genome genes for a protein that allow it to alter UV light into green fluorescence. And the expression of these genes happens in those mantle cells here, in those rim cells, so that the jellyfish would be able to glow greenly, right? And scientists went ahead and actually cut out the gene using a restriction enzyme, generating again the sticky ends and cloning it into a vector that they could use to transform mice. And you see how this works. They cut the vector open with the same restriction enzyme. Now that would allow for this fragment to be inserting itself here by means of those sticky ends attaching to each other. Then you have to anneal the backbone to recreate the sugar phosphate backbones. And what you get ultimately is green fluorescent mice. Now scientists in that sense were very lucky, because the green fluorescent gene from a jellyfish doesn't mean that it would have any activity in mice or other organisms, but it does, right? You see here a normal mouse embryo and a mouse embryo of a mouse that was transformed with the GFP gene, the green fluorescent protein gene. And what you see from there is actually that the glow is really here. It's much more significant. There's not really a fluorescent glow in this embryo, right? That fluorescence is here in the transformed one. And that's actually a technology that's then being used for genetic engineering, as well as for studies. Like here is a study in terms of bone marrow cells, like how would they actually lead to osteoporosis? Are there any regulatory factors in the gene expression that lead to that, right? So we are coming back to our experiment here that we want to do together. We are talking now in our laboratory that we want to do not anymore about bacteriophage T4, but another phage that infects E. coli. It's called bacteriophage Lambda. It's very similar in its setup to the T4 bacterium, with one difference. It doesn't have several tail fibers, there's just one. But you have also the tail tube, you have the head. The head would contain the DNA, and that seems to be almost 50,000 base pairs long. And here you see some electron micrographs of the bacteriophage lambda DNA, the whole bacteriophage lambda. And I want to go now look at with you, and this is a misnomer here. And that's actually not a view of the lambda phage, that's a view of the T4 phage. But regardless, what I want to point out to you here is in both these organisms, lambda as well as T4, the DNA is actually stored in linear form. And only when this DNA is injected into bacteria does it actually form a circle by means of 12 nucleotides here on both ends that are sticky to each other, right? And that's sort of a mechanism to allow for that DNA to be replicated in the bacteria. So here's our worksheet again, and we had already a lot of these answered. So let me go back right to the responses. Because what I want to look into, like where in these different steps that we had identified, actually the restriction enzymes exert their action. And would you agree with me that it's exactly in where the DNA can invade, but the bacteria are able to destroy the invading DNA by means of restriction enzymes? Now, I don't know whether you saw this here. There's a question for extra credit. If you go ahead and design experiments that would confirm or refute any of these ideas and hypotheses that we had up here, how actually immunity can come about, that deserves not just extra credit, that would be extra credit. All right, let's go back to our worksheet and now see what we can do in terms of bioinformatics. The question here is, what determines, well, it's actually, we know that already, right? It's restriction target sequences that determine that. And they were here GAATTC, and for Hindi 3 it was AAGCTT, right? Good, and you know, I have that here in the response sheet. Obviously GAATTC for ECORA1, and for Hindi 3 it would be these sites here, AAGCTT. Now here's a question, and I apologize for jumping back and forth, but are these sites the sites where the restriction enzymes cut solely and entirely determined by the sequence of the DNA, or are there other factors that contribute to selecting the sites where the DNA is digested? And here's the research question, and the question is like, how can we do an experiment to actually prove either one of these, that it's either all the target sequences that determine the cut sites, or that there's something else to it. So here's what I propose we do, that we actually go ahead and find the DNA sequence of the Lambda genome, and then determine where in the Lambda genome are the target sequences, and determine from there how often the Lambda genome would be cut, how many fragments would come out of it, and how big these fragments are supposed to be. So we're basically building a set of predictions using just the Lambda genome DNA sequence, if we ever get our hands on it, right? And then, in the second step, we would actually do a wet laboratory. In a test tube, mix the Lambda DNA with the ECORA1 restriction enzyme, and then run this out on an agarose gel to see what fragments are actually there, and then compare the results of the wet lab with our predictions. If our predictions meet exactly what is being shown in the wet lab, well, then the answer is a yes. If our wet lab, however, shows a different pattern than what we find if we only look at the nucleotides, then we know, no, there is something else going on as far as directing the proteins to the restriction sites. Cool? So let's get that Lambda genome. And what I want to do is I want to go to the NCBI website here. You can Google for it, or if you have the worksheet open, you can copy and paste and go to a browser. And here's our DNA live schedule and all that kind of stuff. Let's not even go there right now. I want to go right away into the National Center for Biotechnology Information, which is NCBI. Okay, so here we are. So this website has a huge amount of stuff because, believe me or not, this website is not only there for students and for educators, it is there for the entire world, but with a focus on research scientists. Research scientists that want to go into the DNA of many organisms, of many different organisms, and conduct their research. However, it is available for free for us as well. And again, we're not going to right now look into any of these features that are being provided. If you want to join me next Monday, I'm going to conduct a bioinformatics workshop on the true cause of sickle cell anemia. I think I'm going to come back to this site there as well. But right now, I'm going to stick with this simple search tool up here. And what I want to get my hands on is the nucleotide sequence of Phage Lambda. And in that worksheet, you find already a sophisticated search term for that, where we don't just say Lambda genome. We want the complete Lambda genome. And it's a Phage Lambda. And so we're narrowing by adding more and more terms to it. We're narrowing the results down. You know, I'm a lazy guy. I just don't want to have a whole lot of results to wade through. And the icing on the cake here is the Enterobacteria phage lambda. It's a virus, you know, a phage that actually infects bacteria entero that live in our guts. All right, let's search. The search comes back with a whole bunch of items here. Again, lots of features available, especially for researchers. We're going to focus just straight here on the items that were found and click on the first one. Because by narrowing down the search terms that much, we are right where we're supposed to be. And we find a data sheet in the NCBI database that's called GenBank. You see that up here, right? And at first you can see there's actually a locus identified, which basically means it's an identification number that's being used in GenBank to uniquely identify the data that are in this entry. And then we're talking about 48,502 base pairs. I don't know whether you remember from a previous slide that this is the Lambda genome. And we talk about linear DNA that we talked about as well, that it's the linear DNA stretch of the Lambda genome. Very cool. So where are the nucleotides? Well, at first we find a whole lot of references to publications that were published by authors that have worked on the Lambda genome. Let's see how far that goes. Let's grab that. Let's grab the scroll bar here and scroll down. Ooh, a lot of people worked on that Lambda genome. Then here is this. Are there any nucleotides here? It gives us features, right? But as far as I can see, still no nucleotides here. MEV and KK, that doesn't refer to nucleotides. A, C, T's, and G's are what we're looking for, right? But you may remember that one slide about the genetic code. Oh, this relates to proteins. We come back to this in just a short moment. But let's keep scrolling. Ooh, a lot of proteins in that phage DNA genome. And A, C, T's, and G's. Starting with nucleotide number 1 is a G. Okay, sequence starts with GGG. Where's the rest of it? Well, let's just keep scrolling and see what we find. Oh, the Lambda genome stops at ACG. And is how long? How many bases? Well, 48,481, 48,491, 48,501, 48,502. Oh, we are in business. That's exactly the nucleotide sequence we're looking for. Let's just go back to the beginning of that nucleotide sequence, and you see how it ends here in ACG, right? Ends here in ACG. And let's go to the beginning. Remember those GGGs, here the nucleotide sequence where it starts. And there's something baffling. Let me ask you a question. In the Lambda genome, do you think there is a break here between this C and this C? A break between TT, a break? There is even a line break in the Lambda genome? Well, you guessed right. Absolutely not. In the Lambda genome, this C is directly connected with a sugar phosphate connection to this C. And so this T to this T. And ultimately, this G doesn't have any numbers between this G and this T here, but it's a sugar phosphate connection between this G and this T, right? Okay. Numbers. Do you think there are any numbers in the phage genome? Nope. There are nucleotides. These are molecules, and that we are abbreviating here in the database with A, C, T, and G, right? But when it comes down to it, it's sugar rings and side chains and all that kind of stuff. So there are neither letters actually nor numbers in the phage genome, right? Number 3, what would you expect to pair with G? Yep, C pairs with G, and so G pairs with C. Now, but there's a T written here. What's going on? Well, the database doesn't display double strands. The data displays single strands, so that the sequence goes to this G and then continues right here without numbers, of course, without breaks. But this is a single sequence. So the database saves actually space by not displaying both strands. Why is this okay? Well, it is okay because once you have one strand, you can easily determine the other. Because where there's a G in one, there's a C in the other. Where there's an A in one, there's a T in the other, right? So not a big deal, and actually saves a lot of storage space if the National Center for Biotechnology Information doesn't have to store both strands for each sequence, right? Okay, cool. Let's just see what our worksheet wants us to do. Our worksheet asks us, how big it is. Well, that's the 48,500. And then it says, well, why don't you look a little bit in that features listing for the first gene that's listed in there, okay? Let's do this here real quick. We saw features already on our way down. And let's continue this here by going to the beginning of features. I could have probably just searched for features. And it tells us, there is a sequence from 1 through 48,502, right? Which is down at the bottom. And in that sequence, arriving in nucleotide 191, that's where a gene starts. And that gene has a name, NU1. It extends all the way from 191 to 736 and encodes a gene product, which is a DNA packaging protein, right? In some organisms, there's a difference between gene and coding sequence. So that's why NCBI always lists these two things. But for phage genomes, from what I've seen so far, it's generally the same thing. So it's basically a repeat here. But then when it comes down to that, we going to the protein product, it's a DNA packaging protein, and the amino acid sequence of that protein is down here, right? The amino acid sequence for that protein is down here. How big a protein is it? Well, let's see how many amino acids it holds. I'm going to copy this out. I'm going to copy this out and count this. And nope, I don't want to count this myself. I'm going to go here and open a new window. And I use Word to help me counting, right? I paste the sequence in here, the amino acid sequence. What's happening with my mouse? Let's hit Control V. I don't know what happened with my mouse. And now I have the amino acids in here. And Word has a feature. If I go to Review, Word Count, it tells me that this amino acid sequence is 181 amino acids long. So it's not a huge protein, but there are some smaller, and so it's on the smallish side. Okay, cool. So what else is in our worksheet? We know now everything about this NU1 gene and the protein. Oh, in terms of determining the length of the gene, you could go two ways. You could either do the math on here. What would be the other way? Once we know the amino acid sequence, right? Remember that was 181 amino acids. That leads to how many different nucleotides? Absolutely right. 181 times 3, because each nucleotide is encoded by one triplet. Now, usually it's one triplet more, so it would be 546, because the stop codon, remember the genetic code table that we looked at? The stop codon doesn't place an amino acid, but it's part of the gene. So, you know, it's some 543, 546 nucleotides that the gene is long to encode those 181 amino acids here. Very much fun. So in the worksheet, there is some more requests for you that you can do later to get a little more familiar with this NCBI website, and basically look at 3 additional proteins. Just search some in that feature sequence that we just looked at, right? Here, the feature sequence, scroll down or whatever you want to do. And then extra credit, and this time it's not extra credit, but just extra credit. Find what is the first letter for the first amino acids in each protein. Is it the same? Is it different ones? And what amino acid is being placed that way? And then for extra credit, I want you to actually figure out 5 more genes from the Lambda genome. And these genes are very specifically a function pertaining to the host cell. So the tip for finding this out is by just searching this web page for host, right? That's all. And you will find the 5 genes that are currently in the database designated as having to do something in terms of the host phage interaction, right? But all these are extra credit questions. All right, now that we found the Lambda DNA, let's see how we can actually use computers to find the different cut sites. And I'm back in the worksheet here, and I'm just going to go down. And the first thing we could do is write actually the sites into our tables here, and VCTT. So these were the restriction enzymes that we found for ECORA1 and HinD3. And one of the first things we could try without any fuss, using actually the browser that we just found the sequence in, and this is right here, to see whether we could actually just search it, Control F for searching. You see this on the lower left-hand corner, and paste the sequence in there, and certainly not with the symbol between G and A, but so we get one hit here already, 1, 2, and that's it. With ECORA1, we get 2 hits. I go back to the worksheet and I write the hits in, 2 hits. Now, if we digest a ribbon twice, how many fragments are we getting of that? Exactly right, 2 cuts, 3 fragments. Fragment sizes, well, they're a little bit more. The first one is going 26, 101, 102, 103. The first one is going all the way up to 26, 104. So it must be 26, 104 bases long, right? Where's our worksheet here? 26, 104. The second fragment, we probably need a calculator for that one, and I have currently none open. So let me go back here and then pull a calculator in, because we basically have to go to the next cut site in here. We have to go to the next cut site, which is at 44, 9, 41, 51, 61, 71, 72. 44, 9, 72. 44, 9, 72. So 44, 9, 72 minus, what was the first cut again? 26, 104 minus 26, 104. And we would arrive at 18,868. 18,868. And finally, the last fragment would then be basically, if we empty that calculator, 48,502 minus 26,104 minus 18,868, 3,530. So that would be the third fragment, 3,530. So for HinD3, we have a different cut target sequence. I'm going to copy this out again, copy this out, bring this back in our browser, and try to find that one. And again, I have to remove this. I don't know what this sign is called, but regardless. So let's see where we find the sites. We find one here at 44,101 and another one at 36,841. Any others? Nope. We find 2 sites again. 2 sites, 3 fragments. And what I'm going to do now is I'm not going to determine the sizes of those fragments. I'm waiting for the next tool that we're going to use to actually determine the sizes for the HinD3 fragments, if that's okay with you all. Okay, so I move forward. We have now an idea that we have 2 sites, 2 hits, 2 cuts with ECORA1 in the sequence using the browser. The next tool I want to work with you on is actually one that many of you probably have used before. I want to see whether we can actually use Word to do this restriction analysis on Lambda DNA. That means, though, that I need to somehow get that Lambda DNA out of this website into Word. And I'm going to go all the way up again, and the instructions are in your worksheet, where it says click this FASTA link, which actually reformats the sequence, taking out all the breaks and all the numbers. There's still a lot of verbiage here, so I'm going to go a little further and take actually this whole sequence in this text window. And now I can take it all out of here, and I keep scrolling with my mouse. I basically hold that cursor pressed and go to the end of it. And now I take this, I copy it, and I said I'm going to use a Word document. And why not using that Word document that we had up before? I'm going to select everything, delete it, so clean it out, paste that sequence in here, and it's ending with ACG. And if I scroll to the top, I have our GGG also in there. Cool. So I have the entire sequence here, and then I can do, Word allows me to do this. I'm using Control F shortcut here to actually type in the Hindi. Oops, sorry, we don't have it anymore. Let's start with E. coli again. Now I forgot what the E. coli recognition sequence is. GAATTC. So let's write GAATTC. And that didn't go too well. Don't know what opened up here. So let's take GAATTC and search for it. I find it once. And actually I find 5 hits in here. 1, 2, 3, 4, 5. Let's do what a restriction enzyme would do when it encounters any of those sites, right? It counters any of those sites in the phage DNA after the phage has invaded. And I'm cutting them all out here like this. I'm cutting them all out here like this. Cool. Now, going back here, that means if we have 5 hits in there, 5 cuts, that means we get 6 fragments. And not the 2 and 3 that we had before, right? Yeah. And I'm going to go and determine the fragment sizes now, and using another feature that Word provides for that. First, I am going to make this whole view much, much smaller. Let's say I put it to 50%. Okay, now I can't read the nucleotides anymore, but I don't have to, right? I just go to the beginning. I just go all the way up to the beginning and highlight what was the first fragment. I don't take that first line because the first line has a description of its lambda and all that kind of stuff. But here is our first fragment, right? And then Word has a feature under review that allows you actually to count characters. And that first fragment is 21226. And I write it in my worksheet. If I close this first, write it in my worksheet, 21226, right? And I do this for all 6 fragments. I'm going to highlight the next fragment here. And I am going to review word count and find out it's 4878, 4878, 4878. And let's just do the prudent thing here. Okay, let's see the next fragment. We would continue down here into this end of this fragment, right? And I know a shortcut, Alt-T-W, gives me the counts, 46, 50, 43, 56, 43. Oops, let's do the prudent here, 56, 43. And let's see the next fragment. Let's see the next fragment here. And we move into this one. And I am missing that first stretch. Can you see that? I have to go back to this and now do this again. Alt-T-W. It gives me 7421, 7421, 7421. And let's take the last fragment. And Alt-T-W, the pre-last fragment is 5804. Close and 5804. I hope I highlighted correctly, but let's just assume for now that I did. And then the last fragment that we have here is actually this one here, and I'll do it one more the pedestrian way and do the word count 3530. Oh, for 3530, that's actually a fragment size that we had seen before. I don't know whether you remember, but even the search in the browser gave out that fragment correctly, right? Now I could do the same now for Hindi, and I will do the same for Hindi as far as the search in the Word document of AAGCTT is concerned. And I'm going to go back to this view here and open this, copy this into a new Word document. I copy the sequence into a new Word document, new blank document. I copy the entire sequence in there. And now I'm going to go back to the regular zoom that we had before. And that was actually 160%. Not sure that you remember. Okay. And I go all the way to the beginning. All the way to the beginning. No, what's happening? Oh, it's still searching. Yeah, okay. All the way back to the beginning. And from here, I search Control F. I need the sequence AAGCTD. And I'm going to search for that sequence. Now, for some reason, it wrote actually the sequence into the document. That's not what I was trying to achieve. So 1, 2, 3, 4, 5, 6 cut sites. And in this case, I'm not going to simulate the cuts. I'm just going to write those results in here, 6, 7. And I save myself also to write the sizes in here. Why is that? Well, because we have 2 more tools that we could work with. And let's take the last one to determine the E. coli sizes. And then you can always go back and double check with what we found in Word and the others if you want to. Okay, so one would be GeneBoy, and I'm saving myself this right now just because I'm not sure that you all have Adobe Flash. It's a fantastic tool, but what I'm going to use instead is actually this that's called Clipper, seemingly even in its name being tailored to digesting, clipping DNA sequences. So I'll do this here. I call up that Clipper. There's our sequence Clipper. I have to go back and get my Phage Lambda DNA into it. Phage Lambda DNA into it, into that Clipper. I paste it in here. I'm giving it in the restriction enzyme, echo R1. Oh, that wasn't very smart because I had all caps on here. Echo R1, and I run this. So let me move this video window out of the way. And so we have our sequence back here. And in fact, we have our 48,502 nucleotides up here. But now what we get here on the right-hand side, that it actually is being, the DNA is cut in 1, 2, 3, 4, 5 sites, generating 1, 2, 3, 4, 5, 6 fragments. Let's enter that into the worksheet. So it's cut at 5 sites, generating 6 fragments. And the fragment sizes are as we have them in here. So I can really just take them out, Control copy, or let's do it the way that you can see what I'm doing. Copy this out into the worksheet. And in here, I would like to paste it in a very specific way, but as text and not HTML. And now I have it in here. Now I can basically replace all these with a semicolon if I wanted to. But you know what? I saved myself that hassle. And let's compare those sizes. 21 to 26, yep, we have it. 48, 78, yep, we have it. 50, 72, and we have that, and we have that. So Clipper got us everything in a much shorter time frame, right? So let's hope that then is also true for the Hindi 3 experiment. So let's use, let's do that digest using Clipper again for the Hindi 3 fragment and not having to determine now our restriction fragments by hand. So what I need to do is I need to go back to this one here, and instead of equal 1, I write in there Hindi 3. Very cool. So I get the information that there is 1, 2, 3, 4, 5, 6 restriction sites leading to 1, 2, 3, 4, 5, 6, 7 fragments here. 6 restriction sites, 7 fragments. Let's write this in here into the worksheet. 6 sites, 7 fragments, and there are our sites. Let's do this again. Just paste it in here. So as I said, we have saved us this work before, but at least in the Word document, we had 6 sites and 7 fragments, as we have here. And if you were to do the rather pedestrian way in Word to determine the sizes of the fragment, you would see that exactly these fragments would come out. All right. There's one last thing that we have to look in here, because the beauty of this tool, of the Clipper, is that's a dedicated tool, right, to actually conduct restriction analysis on DNA. The beauty of it is that it's really tailored to that task, and therefore allowed the programmer to do a few more things that you wouldn't find ever in Word or if you search a browser. And that is, the programmer went ahead instead of just giving us the sequence of the fragments as they are in the Lambda genome. He said, well, what if you would take all these fragments, would put them in a tube and line them up by size, in an agarose gel, say. Then the smallest fragment would run fastest and furthest, and the largest fragment would run the slowest, right? And then you would get actually a differentiation and separation by size. And therefore, we get actually a prediction here what the agarose gel would look like if this were in a wet lab. And if the restriction cuts are determined only by the nucleotide sequence. You ready to check that out? Good, because in our shared Google Drive, we have actually a gel here that we can look up. So first, 1, 2, 3, 4, 5. 5 restriction sites for EcoR1, 5 fragments for EcoR1. Oh, it looks like 5 fragments, right? 1, 2, 3, 4, 5, 6, 7 for Hindi 3. What's going on? We should have actually 6 fragments in EcoR1. Well, here is what's going on. See our echo digest? These two, 5804 and 5643, run so tightly, are so close to each other, just 150 base pairs apart from each other, that if you were to look at them, in an agarose gel, like we do here, this thick band, you might even be able to see, this is much more DNA than should be only in one band, right? This thick band contains both of these fragments. So if we let this gel run longer, we would lose this small 564 base pair fragment, but these two bands, this large band here would actually separate out into 2 separate bands. That would be the equivalent of 5643 and 5804. So let's see whether that was exactly what we were looking at. See here, 5643 and 5804. So what we find then is that the agarose gel at the wet laboratory actually gives us the same results as the bioinformatics tool. And therefore, there is nothing extra that determines where the restriction enzymes cut than just the target sequence. There's nothing extra. Otherwise, we would expect the difference between getting all these predictions done, just dealing with the sequence, and then conducting the experiments using the real enzyme and the real DNA. Instead, they gave us back exactly the same things. We didn't determine exactly the size of the fragments in the gel here, but at least the fact that we only have those 7 fragments here, that we have 6 fragments here, that we have 2 that run very close together here, that we have 2 that run very closely together in the Hindi 3. See, here's the Hindi 3 digest. Those 2 run very closely together, but as they are further down in the gel, they separate out better than those 5643 and 5804 fragments, right? So we get the same results with the wet lab as we are, sorry about that, need to go all the way back, in the wet lab, as we are getting them with our electronic restriction analysis. And therefore, our question whether there's anything else that directs the restriction sites is a no. It's only the restriction sites that determine where the DNA would be cut. So you all did very well holding up to now. And now I want to go ahead and show you how in that tool there is actually an extra credit question again. And here it is. And let me show you the responses first, just so that you can see them. Responses, in the responses, you would then write that you would enter all those fragments in and all that kind of stuff in the tables. And then under conclusion, the answer to the question whether only the nucleotide sequence determines where the enzymes cut is yes, right? Because the wet lab confirmed the sequence-based predictions for the number of the cuts and the fragments and the fragment sizes. So here's the extra credit question, and I go back to the worksheet. I want you to develop some ideas for a problem that we have with our model. I don't know whether you realized, but E. coli is synthesizing the EcoR1 protein to destroy the phage DNA as it enters the E. coli cell. But in the E. coli cell is not only phage DNA. In the E. coli cell is also E. coli DNA. However, the E. coli DNA does not get destroyed by the by E. coli's restriction enzymes. It doesn't. So using that approach that we used before, in terms of just thinking about potential scenarios, you know, as we did here, how could a bacterium develop immunity to phage attack? I want you to think about what could actually protect the E. coli DNA from being destroyed by its own restriction enzymes. Thank you again, and I hope I see you next week in the bioinformatics laboratory to determine the true cause of sickle cell anemia.